Sounds good.
Good morning, good afternoon, dear viewers, friends, and colleagues. Welcome to the Innovative Sustainability channel. Today we have our guest, Siddhant Sharma. He's joining us from Dubai at present. He recently completed his bachelor's degree in chemical and biological engineering at University of British Columbia. And we worked together in Sanofi, in my team at that time, via physical and part platform. So Siddhant has both academic and industrial experience. And today he will talk about the methods, analytical methods, and how they're used in vaccines and contribute to sustainability of the product. So Siddhant, it's great to see you on the channel. Thank you for agreeing to join. And
Yeah, thank you so
during your
much, Marina.
vacation time, yeah, I really appreciate
Yeah,
that. Considering, tough journey with, graduation and exams. So
yeah,
welcome, and the floor is yours.
yeah. Thank you so much. And I'm just going to share my screen now. Okay, here we go. Can you see my screen?
Yes, yes, all good. I don't know if you can do it like full screens. Some
Oh, yeah,
speakers
there we
can.
go.
Yeah, very good.
Yeah,
Excellent.
okay.
Thank you.
Yeah.
Thank you.
Okay. So thank you so much, Marina, again for the lovely introduction and for having me here. It's a real honor to work with you again on this presentation. And so today I'm going to be talking about innovative and sustainable analytical methods in vaccine development. So I'm going to discuss like the existing challenges that are there with developing new analytical methods, what solutions there are being developed, and what are some new technologies that are there, and some of my own personal career insights by working in the vaccine industry. As you know, we work together, and so yeah, it's been pretty fun so far. But just for another quick introduction about me, so I graduated from the University of British Columbia this summer, with a Bachelor's of Applied Science in Chemical and Biological Engineering. I have over 5 years of research experience in undergraduate during my undergraduate education, as you mentioned, both in the industry and academia. And I was a former biophysics and PAT intern at Sanofi from 2023 to 2024, and I was on Marina's team at that time. And we worked together towards developing novel biophysical characterization methods for vaccine analytics. And we published one paper there together, and another one is now in has been submitted to the Pharmaceutical Research Journal as well. And so, a quick overview of today's talk. I'm going to first describe the innovations that are happening in the vaccine analytical characterization methods, and then I'm going to discuss the challenges which are there, which are associated with them, and finally discuss some of my own personal career insights for anybody who wants to transition to this industry. And I'm not only going to talk about vaccines, but whatever I'm going to talk about today is broadly applicable to a lot of other biologics as well. So first, let's discuss the overall end-to-end vaccine production process, how it works, before specifically diving into the analytical methods and the innovations that are happening. So the overall the vaccine production process is divided into three parts. One is the discovery, the second is development, and finally is delivery. And this includes all different teams of scientists, engineers, clinicians, you have politicians, and then of course a lot of transportation and public health departments as well. So it starts off with the first process of vaccine discovery. It proceeds towards the preclinical development side, where the target molecule or the target antigen or the antibody type therapy is further studied and optimized to become more potent and safe before finally developing it into a full vaccine or a full treatment. That is the part that occurs in the process development part, and then finally we start off with the manufacturing. It is manufactured, and then it undergoes phase 1 to 3 clinical trials, each being more rigorous than the previous one. So the most rigorous one is the phase 3, where a lot of thousands and thousands of people globally, in fact, are exposed to the vaccine to determine its efficacy and safety. Finally, it achieves the national regulatory approvals by the standards set by the World Health Organization and industries such as the FDA in USA. And recommendations are made to further improve the vaccine and how to nationally roll it out. And finally, the end of the development phase is the actual vaccination. And based on the results from that mass vaccination, further policies are advocated and its effectiveness is determined. It is the cost effect how cost effective the vaccine is. It's analyzed during this phase, and the final phase of delivery is governed by something we call the EPIC, or epidemiology, public health, and impact. So a lot of the public health organizations in all the different countries like Canada, USA, they together come in this last phase, which is the post-vaccination phase here. So this is the overall basic end-to-end vaccine production process. Starts off with the development of the vaccine, then finally manufacture it for large-scale rollouts. Then the clinical trials happen, and then finally the vaccination occurs, and then its effects are monitored globally. So, but now coming to the core theme of today's talk that I have is that what is the core idea of sustainable vaccine development? When we all, I know sustainability has mostly been only associated with like heavy petroleum, oil and gas, or chemical industries.
Exactly.
But what about the vaccine development or the biomanufacturing industry here? So in here, what how I feel about this based on my experience as well is that sustainability in vaccine development is fundamentally about doing more science and more development by using less material in less time and less environmental footprint while keeping or improving the product quality as well. So three tenets. One is to use less materials and frankly healthier materials or more eco-friendly materials, and you do and then you develop a vaccine in lesser time. That means it's more automated, it's much more quicker, and then you use less environmental footprint, like you have more energy efficiency, because to run such massive manufacturing industries you need a lot of energy. And a lot of these biomanufacturing facilities, they generate a lot of biological waste which can be extremely hazardous if not dealt with properly, and a lot of chemical waste as well, so that also adds to the environmental footprint. So the idea is to even produce less waste, and while of course keeping or improving the product quality, because fundamentally any biological therapeutic treatment, whether it be a vaccine or any other biosimilar, it's essentially a war between safety and efficacy, and it's in that tug of war safety always wins, because that's the core idea. And so while doing all of this, like using less materials, less time, less environmental footprint, you still improve the product quality to make it extremely safe for the patient. So now I'm going to discuss in more detail about each of these tenets that I talked about. So the which brings me to the first part of my talk here, which is sustainable analytical methods in vaccine production. What are some of the next generation methods that are being developed or are already being used by a lot of these big manufacturing companies like Amgen, Sanofi, Pfizer? So for me, this is really a four. This idea of sustainable analytical methods are divided into four subparts. The first one being greener analytics, as I said, that we need to use more eco-friendly materials for a lot of the characterization work that goes here for vaccines. So the idea of something that we call GAC or green analytical chemistry is being developed right now, and it's frankly speaking not only developed, this has been a part of a lot of the industries for some time now, and which I will of course discuss in greater detail as I proceed with my talk. The second one is miniaturized and supercritical fluid chromatography. So how are innovations in chromatographic techniques actually fueling greater and faster workflows? And of course miniaturized sample preps. That means instead of large scale sample preparation, can you somehow miniaturize it to make it more efficient and make it more quick? The second part is real-time process analytical technology, which is trying to integrate process analytical tools in vaccine production and which can assist with real-time release testing. I know, Marina, that you have been associated with process analytical technology for quite some time now. I know at Sanofi, where you were instrumental in developing the near-infrared spectroscopy method for real-time PAT testing in these batches. And so I will discuss some other innovations as well, for example, in Raman spectroscopy, and how actually at UBC itself that there have been some pretty good innovations with regards to that. The third point that I want to discuss is data-rich platform methods. That is a new method that has been developed that has also been reviewed, which is the multi-attribute method, and how do you integrate it with LCMS workflows. And so this is actually quite a new novel method, which I will discuss. But the idea is to somehow reduce time and the labor intensive assays into a single assay. So the idea is to combine all the different identity assays into one single workflow and then get all the critical attributes from it, which is very interesting. And the final one is predictive modeling. So trying to use more AI and machine learning to develop better predictive stability models for these vaccines. Because, for example, like vaccines and a lot of other biologics are heavily sensitive to pH and degradation by chemical reactions or temperature. So instead of like doing massive amount of studies in them, like stability studies, for example, that take the course of a couple of months, uh, and are extremely labor intensive, can you somehow use AI and machine learning to somehow predict the degradation conditions before it actually occurs? And how can you optimize your entire biomanufacturing process to account for all those? So these are the four tenets that I'm going to be discussing today. And the first one is the idea of green analytical chemistry. So this is the core tenet of sustainable development when it comes to the fields of chemical engineering or chemistry. So green analytical chemistry essentially is to use more eco-friendly solvents, reduce sample preparation time, reduce energy usage, uh, use of more renewable feedstocks, reduce any side reactions or parasitic reactions that occur. You have to use more safer alternatives for safer chemistry. You can what kind of catalysts can you use, for example biocatalysts, to accelerate the production of certain special molecules, or a lot of other things like that, while while producing lower waste. So if you can somehow prevent waste altogether, or you can figure out better methods to reduce that waste. So these are
Mhm.
all the 12 principles of green analytical chemistry, as I have in this infographic right here. And according to the ACS, which is the American Chemical Society, they actually now even have established metrics like analytical method greenness score to quantify how green a method is with regards to all these 12 principles that I just laid out. And this so using metrics like the AMGS right here, like the analytical method greenness score, is mostly right now an academic endeavor, and industry is also now slowly adopting it with more integration and collaboration between industries and academia. So the advances in GAC with regards to vaccine analytics specifically are, one is to you are for solvent free and micro scale techniques, for example in microfluidics or like solvent free extraction technologies. Green chromatographic methods that I had just briefly touched upon before, like the supercritical fluid chromatography, energy efficient mass spectrometry. So some really cool developments are being made into mass spec technology as well, and real time monitoring using process analytical technologies. So let's discuss the very first technique here, which is with regards to micro extraction techniques. So one is of course the solid phase micro extraction technology. So the idea is to use a solid coating that can selectively partition components from an analyte present inside a matrix onto that solid surface. So if you look at this figure here, here we have a matrix of analytes suspended in a matrix. So think of it like vaccine antigens, perhaps inside a formulation, and then you use a specifically designed solid coating here. And when you put it inside here, the analyte of interest that is there, or the antigen of interest, or any other chemical, for example, of interest that you wish to selectively analyze, that kind of deposits on this coating here, and then you can just simply take out this coating, and you will have the separated analyte of interest here. So this is occasionally used to monitor residual solvents like ethanol or any like host cell proteins or lipid degradation products inside these vaccine formulations. And of course, it's also used to determine adjuvant stability. So this is very good for stability studies and rapid quality control and quality check. And it fits well with the GCMS workflows as well, or LCMS workflows, like the gas chromatography mass spec or the liquid chromatography mass spec, for sensitive impurity profiling, and even as a process analytical technology. Not there yet as a process analytical technology, but we're slowly getting there in terms of the developments. The main issue here, however, is that this, of course, is used widely in academic settings for drug discovery and development, but right now it has not been widely adopted due to certain scalability challenges that are there. But we are like the pharmaceutical industry by nature has been very conservative with
Yes.
regards to adopting new technologies. That has always been an issue, but slowly and steadily with new developments and innovations that are there, this we are shifting from this paradigm, and the pharmaceutical industry is now being more and more open to adopting new technologies. So
Yes, you're
besides the
right.
solid phase
It's
micro.
Yes, you're absolutely right. I just want to maybe highlight that adaptation usually happens when technology perceived stable, both in
Yes,
terms of
absolutely.
commercially available, but also in terms of giving you robust results. You don't get
Yeah.
any deviations or issues because the technology is not refined yet and can give misleading results. So this is that's where conservative approach comes from.
Oh
Sorry
yes,
for
that's
interruption. Yeah,
true.
but
Yeah.
I thought it's
No, no, you're
important to
absolutely
highlight.
right. Yeah, thank you for sharing that insight. That's actually true. But I'm actually glad that that this paradigm is now being changed in the pharmaceutical
Oh,
industry with more and more innovations. They're actually shifting from that conservative approach to a more liberal approach in terms of adopting new technologies. But anyways, coming to the
exactly.
second point here, which is about solid phase micro extraction. So solid phase micro extraction, as I just talked about, uses a solid coating like as the primary medium of extraction, as the name suggests. And liquid phase micro extraction uses solvents, very, very small microliter droplets to selectively absorb the components of interest from a complex matrix. So this is used for micro scale extraction, and it is about 100 to 500 times greener than traditional LLEs, as in like it uses very safe solvents. And now more and more investigation is going towards ionic liquids and deep eutectic solvents for liquid phase micro extraction as well, which is good news. Instead of using very harmful like chemical solvents like 1-octanol or ethyl acetate, you can now switch towards going towards more greener options like ionic liquids, for example, which are quite complex and custom liquids, but they of course are much more greener and much more safer than traditional liquid extraction techniques. So this actually helps with quantifying again impurities or even like short oligomer type molecules or even trace metals that sometimes penetrate into such formulations. And the best part about this technique is that it is highly tunable for polar and ionic species. So if you want to analyze like ions or ionic liquids or certain ionic or polar species inside your formulation, then you can selectively study them or like extract them out. So the common aspect is that both of these techniques are very helpful in achieving the green chemistry principles, in the sense that they reduce organic solvents, waste generation, and processing times, and that they are much fast. But the only issue that currently is there is the scalable challenges. Yeah, Marina, you want to say something?
Yes, I just want to highlight, you said short oligomers, but even metals. And usually for metals, we go for atomic absorption. So you have to send, usually companies, sometimes they have techniques inside, sometimes they send it off to subcontractors, to universities,
Right.
with whoever they have agreements. So it's always time and logistics involved. So if you have one method that actually can also, highlight the content of metals, that's really impressive. That's I
Yes,
wanted to
absolutely.
stress out. Yeah.
I'm very
Sorry.
yeah. No, thank you for sharing that. And yes, you're right that conventionally for trace metal analysis, you send it off to another lab or maybe to another university setting that actually are equipped to figure help analyze that formulation. But now with these new techniques, you can actually do this inside your own lab or inside the actual on-site manufacturing area, which is pretty cool. And
Impressive. Yeah.
so the next techniques that I want to discuss are microfluidics. So now one thing is that microfluidics are not new anymore, as in like microfluidics has been research has been going on for a lot of time now. And they actually were actively being used for example, right now we just came out of the COVID-19 pandemic and the success of the mRNA vaccines that were there. And the safety and the potency of those vaccines is definitely stemming from, or they actually owe it to the proper characterization using microfluidic technology that is there. So Pfizer and BioNTech, the ones that, or even Moderna for that matter, who are the principal formulators of these mRNA vaccines, they actually use microfluidics platforms to analyze the LNP, which is the delivery vehicle for these mRNA vaccines. So to characterize them, nucleic acid analysis, the degrading or the stability profiling of those vaccines was used using microfluidic technology. So there is like microfluidics right now is the hype, I would say. Like it is like a very, very core area of research and at a lot of different universities. So not only is it a university academic endeavor, it has actually transitioned there towards actual industries, like for like pharmaceutical drug development, discovery, characterization. And so this has fueled a lot of industrial collaborations between the industry and academia. So this is something that I am personally very excited about. But microfluidics, more than that, also offer a lot of they also function as high throughput screening platforms. So they actually enable rapid optimization of buffer lipid ratios, adjuvant compatibility, sorting, compartmentalization. It's because they are like they deal with like very small volumes, and they're so rapid that they are extremely fast, so it makes it much more efficient. And actually, as a matter of fact, microfluidics is one of the primary reasons for the spin-off of perhaps one of Canada's most successful biotech startups, which is Abcelera Technology. So they actually use a microfluidics platform for antibody discovery, and that actually came off from UBC as well. So the pretty exciting times for microfluidics
Impressive.
research
Yes,
right now.
yes,
Yeah.
absolutely. You
And so
mentioned
the
microfluidics is not long, but honestly, it was from 2007 then you start hearing about
yes, the early two
microfluidics.
thousands, yeah,
Yes, and then very, very
absolutely.
slowly they start to get in, mostly as a LISA opportunity. But
Mhm.
then you see expansion and huge expansion. You're right, happened during COVID time when it's in as a part of manufacturing. So, and they
Right.
succeeded. Yeah.
And so there's a lot
And
of different so microfluidics not only is being used for like vaccines, but even for like understand. So essentially they help reduce like so they help address the problems of a lot of 2D cell cultures which are there. So actually the one of the main issues why drugs do not really translate into very good treatments from say for example animal models or for example from like labs, so is because the complexities between a two-dimensional like a petri dish versus like the human body, right? There's a world of difference between both of these environments, so a lot of treatments that show success in the lab do not really translate well into actual human trials. It's one of the primary reasons is because a lot of the studies do not take into account the real complexity of the human body when a drug enters there, and the kind of challenges that goes around with it, the biological barriers which are present in the human body. So microfluidics actually has helped go down to that micro or nano level scale which usually the drugs inside our body operate under, and actually help understand a lot of those phenomena. So kind of micro patterning substrates or organoids, or trying to test these drugs inside those models has really been facilitated by microfluidics. So we owe a lot of the innovations in terms of drug discovery as well to the recent advances in microfluidic technology as well. So they basically help reduce reagent use, the waste that is generated because they deal in like a lot of small volumes, like the micro or even pico level. That's 10 to the negative 12th of a liter, and so they help with large scale design experiments as well. So you can analyze a lot the effects of a lot of different factors together inside one integrated system. And the other side of of microfluidics or nanofluidics, as whichever name you would like to address it with, as also on the chip, the phenomena of on the chip potency assays. So like you can redesign organs on chips now. You can integrate entire labs on chips, like liver on a chip or like heart on a chip technology. So something like that has been done for analytical tests as well. So something that we call ELISA on a chip. So traditional ELISAs like sandwich ELISAs usually rely on in an actual lab. You would have to use the appropriate antibodies that stick to it. It comes with a lot of like washing steps and purification, and then analyzing it using colorimetric assays. That takes a lot of machines, costs, and expensive production of say antibodies and sample volumes. But by using on the chip devices like ELISA on a chip, you can achieve probably the same sensitivity, but it's 10 to 50 times faster and uses 50 to 100 times lower sample volumes. And so you are being more efficient in terms of time, and you're using less volume sample volume as well, and so you're not generating enough waste as well. So very good in terms of sustain if we are talking about sustainability as a core tenet of vaccine analytics. And so these technologies also, microfluidics is being continuously used in the industry right now as well, in a lot of well-funded labs that are out there. But as time is going to go by, and then as this technology becomes much more economical, we definitely will see more and more current assays which are there being replaced with actual microfluidics work. So excited
true,
about that
true.
as well.
It's interesting you mentioned about organ on the chip or microfluidics with organ. It's something that here in UAE there is also development, and they're thinking about the kidney, on the chip,
Yes.
and Yes, so the growing mainly to assess the effect of drugs, and use the like 3D tissues as the option, instead of going into full body assessment, meaning like animal studies and so forth. So there is a strong drive to shift to that human 3D organs, or
Yes.
organelles they call it, I think it's and assess it with microfluidics, merge it with microfluidics. So there's a development here. You hear a lot about
Yes.
it at the conferences. Yeah. So thank you.
Yes, absolutely. No, even in Canada, Canada, there have been a lot of fantastic developments. A lot of the professors are academically pursuing this and developing next gen microfluidics technologies in top universities like University of Toronto and even University of British Columbia here. So now I'm going to talk about the chromatographic side of it. So right now I've talked about extraction techniques and basic analysis techniques, but now I'm going to talk about purification, like green chromatography. As I had mentioned before, one of the tenets of sustainable development in analytics is newer developments that are being done in in chromatography. So green chromatography is how we call it. So there's three types of chromatographies over here that I want to highlight today. One is the supercritical fluid chromatography, or the SFC. Now, SFC is currently in use for PFAS detection, which is perfluoroalkyl substances in water, which are extremely hazardous to human health. But they're also found in a lot of common things that we use daily. But they found their way in a lot of mainstream water streams, and so supercritical fluid chromatography was developed, not primarily for that, but it has found good application for PFAS detection. But now it is slowly moving towards biomanufacturing and vaccine and other biologics analysis. So the principle technique is very similar to a regular chromatography, except the only difference is that it uses supercritical CO2, which is a very special form of CO2 that exists, um, and it that is used as the primary mobile phase in here. So your samples are suspended in supercritical carbon dioxide, and then it is flown through a chromatographic column, and that helps achieve good separation between different components in
Quick-
a biologics.
Just quick question. So you mean the liquid as CO2, right? It's liquid
Yes,
CO2.
yes,
Okay,
yes, along those
that's
lines.
right.
Yeah.
Got
Yeah.
you. Okay, yeah, it's impressive.
And so this is very good for lipid nanoparticles. Again, the composition analysis of them. For example, if you have certain special surfactants or chemicals present inside the formulation, so to selectively separate them out, or even separating out like different biological samples inside the same formulation. So supercritical fluid chromatography, yes, is currently in use, but not the mainstream right now, which is there. So a lot of the mainstream vaccine manufacturing processes still rely on LCMS technologies, which are currently the gold standards for any proteomics analysis. But slowly and steadily, this new technology, I'm sure, will also find its way towards the biomanufacturing side. So it allows for faster and cleaner workflows in quality control. So not really towards the development side, but perhaps towards the final quality attributes or the quality assurance or quality control side of it. So QA/QC members can be trained to use this for quickly check the sample quality and if
Yes.
it meets the established safety standards. The other
Especially if
one
it is
is
adjacent to the shop floor, any labs that are adjacent to shop floor, they can assess
yes,
very quickly whether the process goes as expected and the product in the condition
absolutely.
that you. Yeah, so this is great. Mhm.
Yeah. And then the second one is the miniaturized liquid chromatography. So essentially the same idea as a microfluidic, like you take a bigger process and kind of condense it down into on the chip type devices. So you essentially use micro scale columns and you use very small flow rates. And this is also currently an academic endeavor right now. And the advantage that it offers is that you can combine it easily with mass spec technologies, just as you would with regular liquid chromatographies. And this can be used for detecting host cell proteins or double stranded RNAs or even other impurities like lipid impurities present inside formulations. And so by miniaturizing it, essentially you use less of the reagent, you generate less waste, you use smaller sample volumes, and so it really helps with making the workflow much more efficient and by using much smaller volumes to begin with and much lower flow rates as well. So you can get enhanced separation using that. And the final is actually aqueous chromatography. So essentially, unlike the first case that I discussed of the supercritical fluid chromatography, you essentially use water or water compatible mobile phases, like simple green and mobile phases in this case for transferring the molecules through the chromatographic column. And this can help resolve nucleotides, mRNA fragments, sugars, excipient impurities or buffers. And just like the previous two, it offers, like, because you're using water rich or like water compatible mobile phases, so you're essentially using very safe solvents here, so there's essentially no toxicity. And it can help with easier disposal in QC labs, so you do not have to undergo those intense or complicated purification or disposal procedures when you're not even generating enough waste to begin with. So this is something that is on the way right now to being adopted by the mainstream pharmaceutical industry.
That's
So
great.
now we're
Yes.
going to talk about, yeah, so now I spoke about the chromatography technologies, but the other component will be the mass spec for actually analyzing those components. And so energy efficient mass spectrometry technologies are being developed. So the first one is called DART MS, which essentially stands for data analysis in real time MS. So essentially, you have a DART ion source, which is bombarded onto a sample on the sample holder, which contains your sample, and that is then analyzed using the mass spec meter. And essentially the core tenet here is that it uses ambient ionization, so with minimal to no sample prep. So in a lot of mass spec workflows, you have a lot of complex sample preparation that needs to be done before you actually analyze the sample using mass spec. And so this essentially offers no sample prep and ambient ionization. So it's very, very ideal for monitoring solvents, lipids, stabilizers, and even process reagents without actually using chromatography. So you probably don't need to separate out your components
Mm-hmm.
before you actually analyze them. So this is hence it's called, and you can analyze this in real time. So the signals are actively being analyzed in real times by next generation softwares which are developed, and hence the word real time in this. So of course, it offers ultra fast, sustainable checks during QC and QAs. And the second one is it's called DESI MS, or the desorption electrospray ionization mass spec technology. And essentially, the idea here is again pretty much no sample prep, and you use specially charged droplets that are sprayed onto a sample here, as it's there in the picture. And these droplets help desorb that sample from the surface, and then it is immediately sent off to the mass spec inlet for analyzing it. So you can, it's essentially instantaneous almost. So you, as soon as you bombard the droplets onto the surface, the analyte of interest is absorbed by the droplet, and the droplet is then immediately sent off to the mass spec unit. So essentially no sample prep, lower volumes, real time and instantaneous. So it offers studying adjuvant dispersion or excipient localization and impurities with like in an extremely fast way. So it supports green analytics by reducing solvents, multi-step preparation, any complex sample preparation steps that need to go in, and make it very, very quick as a workflow. Yeah, you had a question?
Just very quick. Yes. What about footprint? Is it large instrumentation, especially like real data, real-time MS, appealing name?
Oh, yes, actually, yes, this is going to be a big piece of equipment that is there, and an expensive piece of equipment, I must add as well. But the kind of benefits that it offers, I think it is a very, very good investment to have. It essentially all depends on the formulation or the specific biologic that is under study here. And so you can use this both for actual large scale manufacturing in real industries or just for academic purposes as well in labs. Right now, I know that this is used in both, actually, in both the industry and the labs as well. So this is one of those technologies that is kind of has made its way into both spheres.
Thank you.
Now we're discussing about process analytical technology, and I know, Marina, this is like your favorite part as
Yeah,
well.
to the
Like
heart.
yeah, you have considerable expertise in process analytical technology. So essentially, the idea is to how conventional analysis in vaccines or a lot of these biomanufacturing facilities works is that inside a bioreactor, as you produce your product of interest, you then take up a sample and then it is shipped off offline, like to another site maybe, or somewhere else where it is actually analyzed, which we call the offline approach, the traditional reference
Mhm.
approach. But PAT tools can actually help with continuously monitoring critical quality attributes like thermal stability, pH, particle size in both upstream and downstream processing. So the idea is to do it in real time while the process is actually occurring. So you can immediately tell if the process is differing from its desired from its desired trajectory. That is very sustainable because it prevents batch failures, and of course
Mhm.
that prevents thousands of doses which are not wasted. So you save cost, materials, and then you can quickly develop the biologics that is of interest. So besides the offline approach that I discussed, there are three more. One is the at line approach, which is very similar to the